Okay, so this is our second notes on the DNA RNA unit. It's all mutations. Mutations you see here is just a change in genetic material. And these result due to an incorrect base or skipping of a base. And there's other things, but we're kind of looking at these first. What that means is the nitrogen base that makes up the ladder. So we have a DNA molecule, and this is just a part of it. We have the sugar phosphate backbone on both sides. Then you have nitrogen bases attached to it, both sides. And holding it together is a hydrogen bond. It exists. Okay. Then you have, like, let's say A, C, G, C. So on this side you have T, G, C, G, like that. Okay. What happens is incorrect base. If you remember from replication, this splits apart like this. This is the replication fork. And here you have copying taking place. So you have an A, C, G. Here you have a new one coming in, a T, and maybe a G, but now see this isn't right. The G should be a T, and maybe this one becomes a T, and then an A. Okay? Those are mutations. So something happened during the copying process, and the wrong base pair gets put in. Okay? And sometimes they'll fit. Sometimes, though, it gets caught. Like there's little enzymes that are looking for these mistakes. They catch it and stop it and correct it. Okay? But sometimes they don't. And sometimes it may even skip a base. It may not even make the second one. So it may just completely leave it out and skip ahead to the next one. And just make the G. Okay? It's just missing one. You may say, well, who cares? Does it really matter if one base pair or three base pairs are wrong? But if you remember the central dogma, how DNA makes a messenger RNA, which makes a protein, the fact that it's all based on the code. Like, you know, ACT makes a certain protein, and GGG makes a certain protein. Well, if the code's different, it could possibly make a different protein. And that can result in a different trait. And that's really what a mutation is talking about. So that can be an incorrect base, skipping base, or some other things as well. But there's two types of these mutations. The first one we're going to look at is, so gene mutation. This is a change in a gene. Okay? Now, a gene is just a section of DNA that codes for a protein. So it could be, 500 base pairs. It could be 100, 20, whatever it is. But it codes for a specific group of proteins that gets put together and makes something in your body. What can happen, though, is that code gets changed. Okay? It could get changed in like one or a few bases, and they call that a point mutation. And that can be substitutions, insertions, or deletions. So substitutions is where you have an A, and instead of putting a T, it puts a C. Insertions is where there wasn't even one here, and it puts one anyway. So between this A and C here, it just puts another A and a C. Okay? It's just reading the code wrong. Things aren't always perfect, so mistakes can be made. Deletions where it just leaves one out. So the G, maybe it just doesn't do this one at all. Where's the eraser? Maybe it doesn't do this one at all. Just skips ahead to the next one. Okay, and it copies it. And as long as, though, you think about this, as long as it has the original template, that's fine. Okay, but again, this is going to get copied again. Well, the first time it happens, in the end, it might not be that bad. It's when it gets copied the next time, and it has this template that's wrong, and then it gets passed on. Okay, but one can be a point mutation. Okay. And then something called a frame shift can then happen. Okay, a frame shift is a change in the formation of an amino acid, which is just a protein, due to a new base. So then, if you had CCC coding for one specific amino acid, say it becomes CAC, that might code for something else. So this might be tryptophan. I'm not sure what it is, actually. It's something. And this might be glycine. They're different. And that could end up in the protein being very different. Proteins have this different structure. There's primary, secondary, tertiary. The way to understand it is it's like a piece of paper. If you take a piece of paper and you fold it in half, that's, if you take a piece of paper and you, fold it in half, fold it in half, that's like your primary. So you have one protein next to another. If you then take that piece of paper that's folded in half and crumple it up in all these different ways, that's your secondary, okay? Then you take that piece of paper and then you take another piece of paper and connect to it. That can be tertiary and sometimes quaternary. So you have all these different proteins working together, okay, that form the shape of the protein, and they form that shape based on whatever amino acids are found in that protein. So based on what's where, it forms a certain shape. It looks like it does. But if something gets changed, it changes the whole formation of that protein, could, and then results in something being made that's very different than was supposed to be made. A good example is maybe, I'm trying to think, cystic fibrosis, where it's a condition where in the throat or in the respiratory system, proteins aren't formed properly, and it can make people very sick. Okay, there's lots of very dangerous things that can happen when proteins aren't made correctly or frame shift occurs. So if we continue here, you can also have something called a chromosomal mutation. This is a change in the entire chromosome. So this is something that happens many times during mitosis, okay, or meiosis, whatever, meiosis, whichever one you want to think about. But interphase even. And the DNA is copied, and maybe it's done wrong, or maybe during mitosis when the DNA splits apart from the metaphase plate, more than half is pulled to one side, so then on one side you got not enough, on the other side you got too much, and that causes the chromosome to be mutated. Okay? So if we continue here, so we have gene mutation, just a section of the gene, the chromosome, the whole thing changes very drastically. Continue on here, okay? And this can also be, a chromosomal mutation can be a deletion, duplications, inversions, and translocations. Okay, deletion is, if we're going to draw a chromosome, kind of looks like, remember, let me draw this from our mitosis stuff. We have this chromosome kind of looks like that, and this is just the basic one, they can have different types of shapes. But with the deletion, you lose some of it. So if you remember from mitosis, this part is connected. This part is connected. And then it has this kinetochore, and it pulls apart, one this way and that way. Okay, but say this happens. Say this one doesn't come apart. 3 go that way, and then only one piece goes this way. Okay, deletion. And some of it's lost. And then this cell and all the other chromosomes are correct, but if this cell happens to fertilize the egg, it's missing part of the chromosome. That's a deletion, it's a mutation. Okay. Also duplications. When you have extra chromosomes, this is kind of like a duplication, and you have extra. Okay. Because this part right here actually is a duplication of that. And when you have extra alleles and such occurring, it causes a difference in how the traits are expressed. Inversions are kind of like, you see, where things are turned upside down. It's like when you look in a mirror, everything is an opposite. And what happens to the chromosome, it kind of gets changed when it's being copied and such, and it's different now. It's like an inversion or a mirror image of what it was. That also can affect what gets passed on. The last one, translocation, is actually where a piece of the DNA gets moved. So we have, let's say we have this section right here, make it yellow. Actually, get the pen, make it yellow. So here's a section of DNA, and here's another section that's red. Okay. On this chromosome, but they get switched. So this one goes here. This becomes pink, so just color it over. And this one becomes yellow. And if you remember, we're talking about Mendel, that some traits are linked. So then this could affect how other traits are expressed, and that's another type of mutation. Okay, and look here, important part to remember with each one of these, loss or all parts of a chromosome can be very detrimental to how the traits are expressed. And you have extra copies, you have extra alleles, also traits can be expressed differently. And then in reverse in order of how the alleles are arranged, that can result in a different protein being made. And the same thing with a rearrangement of two or more. It also results in a different protein being made. All of these often result in conditions that are lethal, that end up in the organism dying if this gamete if it becomes a gamete, and then it produces offspring. There's a lot of interesting types of conditions that occur, like Turner syndrome. Ones people are really interested in are the X chromosome ones and the Ys, where you have duplication. So instead of having an X and a Y, you have X, XY, or you have X, Y, Y, and that can result in some very different kind of things happening. And encourage you to check those out in your book. But that's our little notes on mutations. Make sure you're checking out the other resources I've given you. Hopefully this is a good unit for you so far, and you don't have too many questions. If you do have some questions, though, let me know, and I'll be glad to answer them. Email is probably the best. You can call, though, anything you want to do. You can send me a message through the course, and hope you guys have a good day. Talk to you later.